Today I will show how to take fluorescence spectroscopy. So this is our instrument. We have a cooling system here. It is working. You can see the UV lights. It goes from the excitation source. From excitation source it goes to the sample part. And we have also emission spectrometer and detector. By the way, detector is cooled by this water cooling system. And the water temperature is right now 5 Celsius, and this is our computer unit. But first I have to put my sample inside the spectrometer. So this is my sample. Detector slit is off. I want to protect the detector, therefore it is in the off position, so there is no light for the detector right now. I will put my sample, and this is my sample. I prepared it in a few minutes ago. So I put my sample, so you can see it is there. There is also some powder that is going off from the sample, but it is not important. And I will close the lid. So I close the lid. I will also cover the surface with this black blanket. So I completely cover the spectrometer. Detector part is a little bit more important, and I will turn on the slits, so right now light can go to the detector part. Then, so this is our computer. Okay, I have to select my configuration, browse, Betamax, layout, and the correct layout for us is this one. I will say OK. So I have to enter the excitation monochromator volume, which is written in here. So it is 330. And by the way, emission monochromator value is 650. So 330 for the excitation and 350 for the emission. So it is correct. I don't have to change. Yes. And applications, I want to take the spectra, experiment, okay, and collect. So this is a TiO2 sample. The excitation should be 330, and it will start to the 40. And scan will end at 650 nanometers. I will take the integration time to collect more signal, and I will change the detector voltage to increase the signal intensity for the sample. And okay, so I should select my data. And I will enter the today's date is 0 02 not 03 25 01. Okay, and I will click OK. So right now, 1 second, there is no filter. I am not using the filter right now. There is no filter in the instrument, and I have to note it into my lab notebook, and I will run. So data is running. So right now you can see the emission spectrometer wavelength value here. It will go to the 340 to start the scan, and this value will be 650 at the end of the experiment. On the other hand, in the excitation part, we have 330, so it is not changing since I adjusted this value, so there is no issue here. And the scan will start, and it started, so it is decreasing right now. And I will show the spectra after a while. So this is our spectra. So if you want to see the peaks, I have to zoom. So it is not easy here, but I can make the zoom a little bit. So as you see, there are peaks. By the way, this is a TiO2 sample, therefore it is not very photoluminescent material, but we can see a few peaks. In order to see the peaks more intense, I have to use a filter, and my filter is simply here, and I will put my filter here. But before putting the filter, first I have to protect my detector, so it should be in the off position, so any light shouldn't go to the detector since there is a lot of light right now. And I will put my filter here. This is 360 nanometer long pass filter, which means that any light shorter than 360 nanometer will not go to the emission spectrometer or the detector. So I will put my filter here, I will cover it, I will turn on my detector, and on the computer side I will click collect, experiment. I will change the last number as 2, and right now there is a filter, and I will say run. So it is preparing, and I will show the data after a while. So it started to run, and counts at the beginning is very small since there was a filter, so this is important. And this is the first peak, excitation peak. So this is the data, so it is pretty different from the first one. And to take the data, by the way, I will just click right, I will copy it to the clipboard, and I will save it on a notepad. Data. So new text document. I will paste it. I will save it, and I can rename it. So that's all.